It is estimated that there are more viruses on our planet than stars in the universe. Less than 1% of these viruses have been studied in the laboratory. We call the other 99% viral dark matter. Now, a lot of these viruses are out in the environment, in the water and the soil. Most of these viruses infect bacteria. But humans also contain complex communities of viruses. Some of these viruses can cause acute or chronic diseases like influenza or HIV. But the full impact of the multitude of viruses in the human body is relatively understudied due to challenges in identifying these small, diverse, self-replicating genetic elements. Characterizing this viral dark matter is only just beginning, but is an essential step in understanding the role of viruses in human health and disease. For my thesis work in the laboratories of Rick Bushman and Ron Coleman, I used high-throughput, unbiased metagenomic approaches to study viruses of the human respiratory tract. In one of our projects, we studied samples collected from lungs of people who received lung transplants. As we were analyzing these samples, we came across an interesting pattern in our metagenomic data. We found little pieces of a virus that had never been seen in a human before. This is actually pretty common in these types of studies. We identify viruses by comparing them to ones that we already know about. But what do you do if the virus doesn't exactly look like anything else? To address this problem, we assembled the little pieces of DNA in the sample and recovered complete circular viral genomes. We believe these genomes were viral because they were small and contained the types of proteins that other viruses are known to have. Now that we knew what we were looking for, we developed a bioinformatics pipeline to search for these new viruses in publicly available metagenomic data. We reasoned that other people might have found these viral genomes in their studies, but didn't recognize what they were because they didn't completely match any known virus. When we did this, we found out that these viruses are only found in humans, primarily in the respiratory tract. We also determined that higher levels of these viruses are associated with gum disease and critical illness. As we found more similar genomes, we realized that this group was unlike any other viral family described before. They really should be their own family, which we called Redondoviridae. Redondo is the Spanish word for round. Currently, we're trying to understand both the molecular biology and epidemiology of redonoviruses. For example, which cells can they affect, and how do they replicate? What human disease states are they associated with, and are they recognized by the immune system? So overall, we use metagenomic approaches to illuminate the vast world of omnipresent viruses and help enhance our ability to interrogate their role in human health and disease. I'm very honored and grateful to have received the 2019 Fraser Prize in Microbiology, acknowledging the importance and impact of this work.